Howdy, folks. Thanks for checking into Mr. Ulrich's Land of Biology. I am Mr. Ulrich. In this video, we're going to be talking about gene linkage. And if any of my students can figure out why this is an appropriate photograph for gene linkage, I don't know, be the first student to send me the title of the movie in the Google Classroom and points. Now, if we're really going to start with a discussion of genetics, we really got to start with our old pal Gregor. Now, we've already talked about Gregor Mendel at length and the three laws and how amazing it is that he was able to figure all this stuff out without knowing anything about DNA, the molecule, or the nucleus, or any of that good stuff. We don't need to go over his laws right now. What we really want to talk about are how his laws, where do they kind of miss the mark, really. A good example of this is the law of independent assortment, which states that upon gamete formation, meiosis, pairs of alleles segregate, right? Pairs of alleles for a trait segregate independently of other pairs of alleles for different traits. What this means is that, say, I have a couple traits, right? Blue brown eyes, or hairy knuckles, not so hairy knuckles. And I have an individual who is heterozygous for both of these traits. The law of independent assortment says that even if you get the dominant brown-eyed allele, yeah, you can get the dominant hairy knuckle allele. If you get the recessive blue-eyed allele, yeah, you can get the recessive normal knuckled allele. But you can also get all of the other combinations because they segregate independently. They all kind of mix up. Now, we know that this isn't necessarily the case. We know that it's not individual alleles that are lining up during meiosis, that it's actually whole homologs, whole homologous chromosomes, pairs of chromosomes are going to segregate. And so it's not 50,000 different packets of information that are segregating independently. It's actually 23 packets of information that are segregating independently from one another. So we know that this law of independent assortment doesn't always work because there are traits that do tend to be inherited together. Take red hair and freckles. If you get red hair, you tend to also have freckles. This means that when genes are linked, they're found on the same chromosome. For a kind of simple example here, I have one chromosome that has all A through E dominant alleles, and on the homolog, it has all the recessive alleles. This means that during meiosis and upon segregation, if the gamete that ended up becoming part of the offspring brought the dominant A allele, it would also contain all of the other dominant alleles that, in this case, because they are found on the same chromosome, law of independent assortment would just not apply. As we're going to see a little bit later, sometimes it'll act like they're segregating independently of one another, but we'll get to that in a second. That's all crossing over. We'll get there. If genes are on separate chromosomes and there's no linkage, then we're totally going to follow the law of independent assortment, and we'll see all of those variations from the heterozygotes. I introduced this concept of crossing over back when we were talking about meiosis, and I got a lot of questions on it because it's kind of weird, and it's really an important concept to understand if we're going to explain how sexual reproduction leads to more variation. And this is an important way of leading variation because this process of crossing over is highly variable. Sometimes it happens and sometimes it doesn't. And it can happen at different points, and so it gets really, really shuffly and wonky. Now, if there isn't crossing over, here's my two individuals that are heterozygous for these two traits. So on one chromosome, they have the dominant alleles for the two traits, and on the other chromosome, the other homolog, they have the recessive traits. And without crossing over, I'm either going to get gametes that have one genotype, just like the parent, right? The parent chromosome, I would say, just like one chromosome, both dominant, or just like the other parent chromosome, both recessive. But once we start to do this crossing over and we get this shifty going on here, and this is going to take place only between non-sister chromatids, and it only really matters when there's a heterozygote situation. If there's not, then it's the same alleles and it doesn't matter what's being shifted, right? So anyway, if there's a heterozygote and there's crossing over between these non-sisters, that's where I start to get these recombinant gametes. These are like not like either of the original donor tetrad here. And so I end up with these recombinant types. This means then that essentially crossing over unlinks linked genes because they're on the same chromosome. Here's a bit more of a straightforward diagram of the same thing, whether there's crossing over or not crossing over. If there isn't crossing over, then I only have those parental genotypes to work with when it comes to the results, to the gametes. It's either like one of the homologous pair or it's like the other. I still get those genotypes here with crossing over. Right, but I also get these other two types where I get in this case, the dominant and the recessive, and in other cases, the recessive and the dominant. But it doesn't matter. The important thing is that it gets shuffled up and that we get different combinations with this crossing over. So how can we test if genes are actually linked or not? Well, of course, I'm going to head to the lab and I'm going to do a bunch of research. In this case, I'm going to do some test crossing. It's important for us to recognize, again, that when we're doing this, we're dealing with stats, so we want big numbers. I'm not just looking at the results of one cross. I'm looking at as many crosses as I can do in whole populations. So I'm going to do kind of like Mendel did. I'm going to start out with a population that is purebred, that is homozygous, dominant for two different traits, in this case, yellow and round peas. I'm going to do the same with the two traits that are recessive, right? Green and wrinkled peas, right? Make sure that I got the homozygous recessive, homozygous dominant. And then I'm going to cross them. And of course, all of the offspring from the cross, the F1 generation, are going to be heterozygous. They're both going to be phenotypically dominant, but carry the recessive alleles. Now, it doesn't matter if they're linked or not. That's going to happen. Now, I can take individuals from that F1 generation and back cross them with homozygous recessive individuals from the same parent population. Right? Now I should get equal amounts. If they're not linked, I should get equal amounts by law of independent assortment. I should get equal amounts of each combination of phenotypes: dominant, dominant, recessive, dominant, dominant, recessive, recessive, recessive. Right? If I don't, if I start getting a skew towards the parental combinations, right, then I know that I have some linkage going on. T.H. Morgan, back in 1933, won the Nobel Prize for his work determining the role of chromosomes in heredity, working with Drosophila. And here are actually the results from one of his experiments following two different traits, in this case, the color of the body. If it's gray, that's normal, that's dominant, and if it's black, that's a mutant. That's the recessive trait. Likewise, with the type of wings, if they have normal wings, which is dominant, or if they had kind of vestigial, kind of barely little wing buds instead of functional wings, that's the result of a recessive allele. And you can see we would expect there to be equal amounts, right, in each group. But actually, I get up here in the 900s in the parental types and only down here closer to 200 here in the recombinant types, the types that aren't exactly like either parent. And this tells us then that there's got to be some sort of linkage going on, right? Because we have the skewage towards the parental types. I think I made up a word. But I do have some crossing over going on. And I can actually calculate like a percentage of this crossing over. So I can get we can call it the recombinant frequency, the amount of crossing over that's taking place. It's really easy to do. All you got to do is add up the recombinants and divide that by the total number of offspring. Right, multiplied by 100, of course, and that'll give you a percentage. Now, so what? We got this percentage. What am I going to do with this 17%? Well, it turns out that the higher the percentage of crossing over, the further apart those two genes are, right? The bigger the chances that there's going to be some crossing over event that takes place in between the two of them. Also includes distance from the centromere, but with this idea, we can actually start to build a map of how close together different linked genes are. As we start looking at more than one allele pair on the same chromosome and looking at the degree of linkage between the different ones, we can start to assemble like an actual map, a sequence of genes. Now, these aren't actual distances, but they are showing relative crossover differences. Now I have a situation where I have to be able to determine if it is independent assortment or if it's not, if it's gene linkage that's taking place. And of course, since the amount of crossing over that can confound gene linkage is going to be variable, there's really one set of data that's going to perfectly exemplify independent assortment, right? Quarter, 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 even ratio, right, of each one of the phenotypes when it comes to the test cross. And if now it sounds like, hey, wait a minute, I have a situation where I have some, expecteds, right, a nice convergent set of expected data, and I have some observes, right, that may or may not diverge from the expecteds, I can kind of figure out statistically if I have gene linkage or not. And if this sounds a lot like using chi-square analysis and goodness of fit, that's exactly where we're heading. But that's going to be in your next assignment. If you have any questions, please feel free to contact me through Google Classroom. And if you're seeing this on YouTube, then post it in the comments. I might see it there, or send me an email. Other than that, I'm really looking forward to seeing you guys in class as soon as possible. Hope everybody's doing well.